the first step in any study of an enzyme is to actually figure out what the function is. And so we can often purify proteins from the cell that we don't actually know what the function is. And so what we need to know, of course, is one, we need to know what the reactants are. So let's say if we have a new enzyme that is just discovered, we need to figure out what it reacts with. And there's a variety of ways we can figure this out, but we'll get to those later on in the course. So let's say we know that a protein reacts in some fashion. And we need to know, of course, the reactants. And then we need to know the products. Then we need to know the stoichiometry for this reaction. So we need to know, for instance, if does this take 2 of A and form 1 of C and 1 of D with addition of B. We need to figure all this out before we can start anything. And of course, this is we're talking about enzymes. I'm implying this is some sort of enzyme catalyzed reaction. So this is the first step. What is the reaction? Or, better stated, what is the function? Okay? And this can take quite a bit of time, actually, first, just to figure out, because if we don't know much about what the function of this, it may be hard to figure out what sort of substrates are involved for the activity, and then even after that, it may be difficult to figure out what the products are. So this is the first step in any enzyme study. After that, we can start studying this. So if we know what our reactants and products are, then now we can do what are called activity assays. And so we discussed this briefly when we were talking about protein purification. And so originally, when we talked about this, what we were using this was to figure out if we knew the function, we could use this to our advantage to figure out where in a protein purification, so which fraction of protein existed. So we could either look at the reactants, we could either look at consumption of reactants, or we could look at formation of the products. Ideally, though, what we want to be able to follow are the products. So what we need is some way, first, is to probe product formation, or in other words, we need to be able to detect product formation. So detect product. And the reason why we prefer to detect product, because there's no guarantee that the reactions of A and B are going to go all to formation of this product. What I'm trying to say here is that you can have side reactions. For instance, A, let's write it like this, A could, instead of being converted by the enzyme to C and D, it could be converted to some other molecule, F. That's a non-enzymatic reaction. Or B could be converted in some side pathway by the same enzyme to form another molecule, H. And so the only way we could find out any activity that is specific to this enzyme, is specific to this reaction, is by following the products in this case. Because if both of the reactants are being consumed by side reactions, then we're not really detecting something that's specific to this enzyme. So ideally, we want to be able to detect the product when we're actually looking at enzyme activities. And there are a number of ways we can do that. And so you've learned about a number of these in previous courses. So for instance, nuclear magnetic resonance, or NMR. Of course, UV-Vis, which we've discussed, absorption spectroscopy. We can use fluorescence. We can use infrared spectroscopy, and a number of other techniques. We're only limited by what's available. And so once we figured out a way to detect the product, then that helps us design the experiments for trying to figure out exactly how the enzyme functions, and then that allows us to correlate structure and function relationships.